the best available systems for neurorehabilitation are ineffective at promoting outcomes that exceed those resulting from the natural course of recovery. I've spent the last 9 days attending in our neuro ICU, and that phrase bothers me a lot. Not the part about me attending in the neuro ICU, but this challenge, which is that when somebody comes in with an acute neurologic injury, whether it be a traumatic brain injury or stroke or any number of other types of acute injury to the brain, we're of course doing everything we can on the clinical side to immediately provide life-saving therapy to reduce the injury that's happening or could continue to happen to the brain or elsewhere in the nervous system. And then hopefully very quickly, we're pivoting to begin to think about recovery. But the state of the science today, and this is nobody's fault, it's just the challenge of the very complex neurorehabilitation science, is that we don't have a lot yet as a field to offer that does better than what would be predicted by the very best outcome, as it says there, considering the natural course of recovery. And we want to change that. So together, we're hoping that we can push the boundaries, together being us here at MGH in the Center for Neurotechnology and Neurorecovery, our colleagues at the MGH Institute for Health Professions and at Spaulding Rehab, to push the boundaries of neurorehabilitation and really improve outcomes for our patients by combining the translational mechanistic neuroscience that has been going on here for so long with rehabilitation science. And I think really importantly from the standpoint of the Spark Award and from the McCance Center is these asterisks at the end, is that one of the challenges we've had is even knowing what are the right ways to measure function, to actually measure the function of somebody either who's able-bodied before any neurologic disease or injury or afterwards, so that we can actually correctly track any of our interventions that might lead to recovery. And this identification of what are the right metrics of success, what are the right metrics of functional performance, even of upper limb use, I think are going to be valuable, even for discovering what are the right indicators for brain health. What is it that we can identify as an easy measure to obtain in anybody that we can then look back at and say, okay, from a brain health standpoint, all is good, or perhaps there's the beginning of a change and perhaps there's an opportunity for intervention. So some of our team is listed here, and the crayon-like drawings on the right, I will freely admit, is there's a limit to how much time one can spend trying to get everybody's head to be exactly the same size. So there are more who are not on the screen, but the team includes, importantly, Teresa Kimberly, co-PI on the Spark Award, who's at the Institute for Health Professions, a PT scientist, very interested in neuroplasticity. David Lin, one of the recent recruits to our faculty, who is here as a resident fellow. I'll tell you more about David in just a little while. And the many departments across the clinical departments across MGH engaged in neurorehabilitation and acute neurologic care, as well as folks at Spaulding, of course, have been involved in rehabilitation for a long time, and our graduate medical education office, which is an important component of part of the Spark Award and the way we've been able to deploy those really valuable funds. So what are we hoping to achieve through this Neurorecovery Collaborative? So first is really multi and interdisciplinary clinical care, which is something that we've been able to make great progress on over the past year. If we look back even 4 years ago, there was almost no footprint of neurologists engaged in the rehabilitation of patients after acute injury. No doubt that there's a lot of research that's happening, no doubt that there's a world-class acute care being provided, and follow-up care, which was largely based upon preventing additional injury or helping to manage the continuation of ongoing progressive disease. But actually being engaged in rehabilitation, trying to improve function after acute injury, is not something that there was a large footprint for neurology, and therefore there wasn't much interaction between the neurologists in the post-acute care period and the other clinical teams engaged in helping somebody to recover from a brain injury, whether that be stroke, infection of the nervous system, or otherwise. So how did we seek to address this? We developed a monthly lecture series, which I'll tell you about, which is slowly but surely going from monthly to every other week. And I guess it's eventually going to become weekly, if I had to guess. The Neurorecovery Fellow, who I'll tell you about a few of them. We've had a couple annual retreats now, one just pre-COVID, and the other one, I guess it's not, we can't quite say post-COVID yet, but another one fairly recently. And everybody engaged to develop, at least from the standpoint of neurorecovery, a learning healthcare system. On the screen is a graphic of one way to think about a learning healthcare system, where every patient, everybody who we're interacting with clinically, certainly is engaged in the process of trying to diagnose and recover from an acute injury, but also to help us learn how can we get the next person that much better? How can we understand their disease process in a way that we can help them to recover function, whether it be communication, mobility, or independence? Here's at the lower right there is part of the team early on engaged in one of the stroke rehabilitation studies, the SMOT study, as it's known up there on the cake, that is part of this collaborative that was launched a few years ago. And you may notice that the cake, if you look carefully, actually said 100 on there. Originally, that was when we recruited our 100th participant to the study that's led by David Lynn. And then I had to Photoshop in the new number, which is 163. So 163 people have been recruited to date on that study in stroke rehabilitation that begins sometimes within 24 hours of the acute admission to MGH for acute stroke. I'll tell you more about that in just a bit. The lecture series that we've been conducting as part of this Spark Award and as part of the funding from the McCance Center and Mass General Neuroscience has brought together a really wonderfully diverse group of speakers, especially from the standpoint of not just their location and the fields that they represent, but across the spectrum of their of career training, from folks who are just getting their PhD or pursuing their PhD through senior investigators. And we've been able to touch in many ways across the spectrum of neurorecovery. I won't read the list that's there. There's some pictures of some of our speakers at the beginning or at the bottom, but this lecture series began, as did many things prior to March of 2020, in a fairly small conference room with a group of closely aligned clinicians and researchers gathering to chat with each other and to hear about some new ongoing science in neurorecovery. And over the past year, this has really blossomed nicely. There's people from around the country that are tuning in to each of these neurorecovery seminars because of the speakers who are bringing in, because of the multidisciplinary discussion that's going on between neurologists and neurosurgeons and occupational therapists and physical therapists and speech and language therapists and physiatrists and neuroengineers and computational neuroscientists. And it's that wonderful synergy that has been seeded with an S by this award that has created the lecture series, and it's also creating all types of opportunities for ongoing collaboration and research related both to brain health and very closely related to neurorecovery. So one of the real, from a budget standpoint, in receiving the Spark Award, we wanted to make sure that we were beginning to train the next generation of neurorecovery clinician scientists. And part of that is by supporting the Neurorecovery Fellowship. So our first Spark-funded Neurorecovery Fellow was Kelly Sloan. Kelly is listed here, was a Hopkins neurology resident. She then joined us for her stroke or vascular neurology fellowship in 2019. She became our second Neurorecovery Fellow, and she also coordinated the first of the Neurorecovery Collaborative retreats, which I'll tell you more about in just a moment. And as you may have heard Akshata mentioning towards the beginning something about a T32 training grant that's being submitted soon. One of the markers of our collective success, not only in this collaborative, but from our hopes to extend the impact of our research, is that our trainees go on to do great things in great places. And wonderfully, Kelly's now an assistant professor of neurology working at the University of Pennsylvania with appointments both in neurology and in rehabilitation medicine. So I think some of what she not only learned but taught us is now being brought to other places, further expanding our opportunities to collaborate with some great institutions. Most recently, our neurorecovery fellow has been Michael Young. And again, just want to thank the McCance Center for the opportunity to support Dr. Young's participation in the Neurorecovery Fellowship. Mike's extraordinary. He got his bachelor's and master's in philosophy. His master's was at Oxford. And he then, thankfully for all of us, I don't know if it was a detour or not, but or if he was lured into medicine, went to medical school and then joined us, really lucky for us, at the Mass General Brigham neurology residency, where it became immediately clear that he was going to continue to be doing great things with his career. We recruited him to become the third Mass General Spaulding Neurorecovery Fellow. And importantly, he's the first solo fellow. As the first two fellows in the neurorecovery program were also doing other fellowships, one in vascular neurology, the other in neurocritical care. But speaking to the potential for neurologists to, again, really engage in brain injury medicine and neurorehabilitation, Michael's sole fellowship is currently and ongoing in neurorecovery. As he was thinking about doing this, Michael's got a long-standing interest and tremendous insight into ethics, biomedical ethics, and specifically neuroethics. And he's already won an NIH brain initiative, that is the Neurotechnology Initiative F32, which is an advanced postdoctoral training fellowship in an ethical approach to detecting covert consciousness. Michael works closely with Dr. Brian Edlow, who, as part of our CNTR, has been really creating an amazing emerging consciousness program and causing us all to rethink how we approach somebody with severe traumatic brain injury, what we believe their prognosis to be, what tools we have available to us to better understand consciousness when the bedside exam can be limited. And Michael's really helping us to think not only about the science of that, but also the ethics of getting some of that information and how that information is shared not only with clinicians and researchers, but also with family members. So over the past year, during his neurorecovery fellowship, Michael's been involved in clinical care and translational neurotechnology research. That clinical care has been going on in several venues, partially in the Mass General Neurorecovery Clinic, in part at Spaulding, the brain injury medicine program, and in the neurorecovery, both inpatient and outpatient clinics. In addition, he's been out at Braintree Rehabilitation, working with Doug Katz, who's an internationally respected neurologist who's engaged in neurorehabilitation. And he's also been at McLean, again, gaining insights into the neuropsychiatric components and some of the cognitively really important components of recovery from brain injury. In addition, he's advancing neuroethics research, helping me, helping Brian, helping Sid Cash, helping us all to think really carefully about how to develop responsibly the new neurotechnologies that we hope in the next few years are going to be deployed to help restore communication, help restore mobility, help restore function for our patients with brain injury. One last slide about Michael that I couldn't help to put in here. This is his publication list from this academic year. When I look at this, it's hard for me not to laugh, as I just did. Here's another underachiever who only has, I think, 7 or 8, I'm losing count here, publications that he's either authored or co-authored in the past 8 or 10 months. I think this speaks well of things that will be going forward. If you ever get the opportunity, if you see that Michael Young is giving a lecture, please tune in. It's one of the most exciting, elucidating, enjoyable experiences that you'll have. He's really, really thoughtful, not only about the science of neurorecovery, but very much so about how we think about what is right, what is wrong, what is moral, what is ethical, as we bring not only some lack of certainty to our prognoses, particularly for people with traumatic brain injury, but in addition, how we should be thinking about using either technologies that are available today or technologies that will soon be available to help our patients. And looking forward, again, thanks to the Spark award. We're really excited that Priya Srikanth, who will soon be graduating from the MGB Neurology Residency, is going to become our 4th Neurorecovery Fellow. She has a long-standing interest in pluripotent stem cells, their application to helping people recover from brain injury, really understanding not only aspects of neural development, but how we can understand changes in circuitry after any types of brain injury, and how our understanding of how stem cells can be applied may be helpful for our patients. So I hope next year to be able to tell you about all the great things that Priya has been doing during her Neurorecovery Fellowship. A little bit more broadly, leaving from our Neurorecovery Fellows, talking just for a moment about this learning healthcare system. We've wonderfully recruited 2 new neurorecovery clinic physician scientists. One who I mentioned before, David Lin, who was our first Neurorecovery Fellow. He, in the past year, has won a VA, that is Department of Veterans Affairs, Career Development Award for his work on stroke rehabilitation. That's work that's being done here, both at Mass General and at the VA in Providence, at the Center for Neurotechnology and Neurorecovery. Let me say that again, at the Center for Neurorestoration and Neurotechnology. I have to make sure that I properly say the name of each center. And also David won one of the 2020 American Academy of Neurology Clinical Research Training Scholarships. Dan Rubin also trained together with David Lin as a neurointensivist, has become the site responsible investigator for our BrainGate research here at MGH. And he too won a this year's, that is the 2021 AN Clinical Research Training Scholarship. Dan's going to be doing some incredible work in intracortical recording, single unit recording in the people that enroll in the BrainGate trial with tetraplegia from either spinal cord injury, brainstem stroke, or ALS, and beginning to do research into decoding speech, which we think is going to be a really exciting opportunity to restore communication in a very intuitive, fluid, natural manner for people who have lost that ability due to either ALS, stroke, or other neurologic disorders. Again, thinking about this learning health system and the collaborative that's been formed as a result of this award. Here's just a few papers from David Lin and a few of the projects that he's been engaged in over the past year. You'll see in the lower left-hand corner a paper on cognitive demands influencing recovery of the upper extremity after stroke, rethinking in the center the journey that our patients are on as they recover from stroke, and how to get the right clinicians and the right technologies to the right patients at the right moment. I'll let you look at the other images that are on the slide there, but just looking in the green box at the upper right at the collaborations that have really formed as a result of this funding between MGH and IHP and Spaulding, the incredible imaging resources available at Martinos, and collaborators outside our immediate institution as listed there. This, I think, is a growing collaborative and collaboration that's focused very much on helping to identify how our patients will get better from acute brain injury, and really creating a new neuroscience and a new understanding of how to apply what we've learned from fundamental human neuroscience to the important goal of recovery. There were about 60 or so people who gathered just over a year ago for our first annual retreat. And please don't read this. The list is there just to point out that we had 20 perfectly timed 4-minute talks. Every single person was amazingly right on 4 minutes, describing each of their research towards improving people who have had a neurologic injury, and doing the science that's going to create new technologies to help people after acute brain injury. After those that 80 exciting minutes, we broke into some small groups, focused on challenges and transitions of care between inpatient and outpatient, inpatient acute and inpatient neurorehabilitation, even extended outpatient care and rehabilitation. What are the challenges and transitions? How can we use neurotechnologies to help, again, in recovery, and how can we collaborate better across the many disciplines that are engaged in helping our patients to recover from neurologic injury? We then predictably by Zoom, had our second Neurorecovery Collaborative just a few weeks ago. And you may see in the background the now standard screenshot of Zoom with many people who were gathered to, again, think about the patient experience. In this case, we heard from a patient who was enrolled in one of our recent trials of vagal nerve stimulation for recovery of stroke. We were part of a multi-center trial. It was led by Teresa Kimberly at IHP, and that was recently published in Lancet. In addition, there were some breakout discussions using some good web-based whiteboarding technology to think again, how do we improve stroke recovery based on everything that we've learned and begun to incorporate over the prior year, and digging in a little bit more to improving the recovery of people from traumatic brain injury and thinking carefully about disorders of consciousness. So to sum up where we've been so far, the Neurorecovery Collaborative has multidisciplinary interprofessional clinical research team centered around a neurorecovery clinic, centered around the training of our neurorecovery fellows, centered around this monthly, becoming even more frequent lecture series with local, international, and national speakers, having an annual retreat, which we really, as everybody else hope, is in person in 2022, and beginning to really emphasize this opportunity to have a true learning healthcare system where every patient who's coming in to see us for acute care can join us in learning more about not just the disorder that they have or the disease or the injury that they may have had, but how we can improve the care for the next patient that we see and advance the science so that we can improve recovery of function after acute neurologic injury. And this is really very much the result of the support of the McCance Center and Mass General Neuroscience. And with that, I believe I can pause, and I'll be happy to answer any questions. Thanks again.
Thank you, Leigh. Please feel free to put your questions in the chat, or you can speak up too if that's what you prefer.
Akshat, are you running? Or you want to answer questions, or should I?
No, please go ahead. I don't see any questions in the chat, but I was just Rudy, go ahead.
Yeah, I was gonna say that's tremendous progress, Leigh. Really, always fantastic stuff. I'm just wondering, just generally in the McCance Center, how can we best merge our, mission at McCance to, provide indicators and interventions for brain health in general before disease? With a system like yours, it was neurorecovery, but what about preempting neurorecovery? Like, how can we use your systems? I know this is a very general, nebulous question, but how can we think about using your systems to think about promoting or maintaining or preserving brain health, rather than waiting to have to recover from brain health?
Yeah, absolutely.
on sickness.
Absolutely. And the three green words, maybe there were four, kind of early on in the slide. At the heart of a lot of this is what should be very simple, but clearly isn't effort to figure out what is the right way to measure neurologic function in somebody who's healthy and somebody who's able-bodied. How does that change from the standpoint of neurorecovery after an injury, and how do we track that recovery? That is one of the biggest challenges, and the best answers for some of those questions are probably not going to come from the inpatient hospitalization technologies that we can deploy, or even the technologies that we deploy from an inpatient rehab standpoint. But what are the portable, electronically available, easily distributable, high impact technologies that show us how well we're moving, how well we're thinking, how well we're speaking, how well we're interacting with each other? And if we can get those types of baseline data, that kind of information, I think not only will it be helpful from our neurorecovery standpoint, but clearly I believe it'll have a really important impact for brain health.
Yeah, I mean, the idea is what are the indicators for the need for neurorecovery, even when you don't know you need it because you're suffering from many strokes or damage, or in other words, we address neurorecovery when it's obvious. and maybe the brain's degenerated quite a bit by then. Can we preempt that? Can we predict the need for, can we have an indicator for the need for neurorecovery earlier on before the brain gets too dysfunctional, I guess is what I'm, whether we could work with you at McCance on that goal, indicators for the need for neurorecovery before it gets too late.
Yeah, there's some emerging research, including some from MGH, that shows very clearly that even for people who are having an acute injury of the type that would immediately result in hospitalization, it's been the years that led up to that and the ability to have a healthy brain, to flip the words around the other way, the health of that brain, the pre-existing resilience of somebody to that injury has a lot to do with their potential for recovery. And even some of the subtle markers of movement and speech that we may be able to detect early as being changed, and somebody who otherwise may not be feeling any of those effects, I think are going to be really important so that we can figure out how to intervene, how to say, hey, there may be an issue here and possibly an issue that we can intervene on. That to me would be really exciting, because even if we're unable to prevent a sudden, especially if we think just about traumatic brain injury, the recovery from that traumatic brain injury, whether it's due to a slip and fall or anything else, is going to be affected in many ways by the pre-existing function of that person, their brain, their nervous system, and their ability to recover. So I can't agree with you more that being able to promote brain health and identify those key markers early on that may be changing, and then to intervene early and in any of the ways that we can are going to provide additional protection against some of the things that we can't predict later on. I don't is that
no,
seem right?
Yeah, it is. And I think the fact that we keep thinking about it is, and working toward that goal is probably most important. Yeah.
Leigh, I have one question, and then we'll switch to our second speaker, Dr. Haoqi-san. But it's really clear that in order to sort of develop the right neurotechnologies, you need to have a mechanism for sort of robust tech assessment and synergies with other disciplines like engineering and computer science. Could you just comment very briefly about sort of how you feel like that should, how you've been successful in doing that, in bridging those gaps between these different disciplines in order to do this?
Getting a group of neuroengineers and computational neuroscience and scientists and neurologists and physical therapists, occupational therapists all around the table. If you ever have a chance to do that and then you listen to them for a while, we will all use roughly the same words, and they will reliably have completely different meanings. One way to think about that is just thinking about the great work that Michael Young is doing. When we have a neuroroboticist or a soft robotics engineer sitting at the table with a neuroethicist, and they're both talking about autonomy, they are barely talking about the same thing, but they're both relevant to the research that they're doing together. So one of the both wonderful opportunities and challenges over the years has been to get the group of people together to repeatedly have the same conversations, to just understand the perspective that they have on brain health, to understand the perspective that they have on brain injury and on recovery, on the tools that are available to recover, and whether somebody is thinking about when is the right time for recovery, what are the right metrics of success. Some people would prefer to think about how quickly a pig can be moved from one location to another on a board as a sign of success. Somebody else in recovery, for example, from stroke affecting the upper extremity. Somebody else may be far more interested in whether that person is able to reach and grasp, and yet somebody else may be interested in whether that person feels that their own ability whether it be with the affected hand or the unaffected hand, is sufficient for them to be able to conduct their activities of daily living. All three of those are important, but if those three outcomes or metrics of success are being pursued in parallel, that's probably not nearly as valuable as those three people thinking together, what are the interventions, what are the, whether they be the exercises or the actual medical interventions that can be provided that would stimulate all three of those kind of paths to success. So it's having a group, an open group, and an honest, transparent group that says, I really don't understand what you just said, even though I know each of the words that you just used. And that's been not only a lot of fun, but I think has allowed us to provide better care to our patients and hopefully to be able to prevent some of these injuries to the nervous system as we move forward.
We're going to move on to our second speaker, Dr. Haoqi-san. And Haoqi, while you're setting up, Brandon, I'm going to hand it over to you so you can officially introduce him.
Sure, yeah, it's my pleasure. So I've been working with Haoqi since I think 2017, when he finished his PhD at NTU in Singapore in machine learning and signal processing. And actually, Matt Bianchi and I started working with him back then, and we just loved it from the beginning because he was always willing to tell us when our ideas were wrong, and he always had his own ideas. It's been really great. So he's been doing work in my group over the last several years that's about what we call the sleep brain age index, and I think he'll tell you about some important extensions of that. And in a couple of ways, this is a bridge, I think, between what Leigh talked about in this acute setting, and then what Rudy was asking about. How could we know in advance, who might do well and who might not do well, and maybe with enough advanced warning, maybe we could help people be more resilient. So yeah, turn it over to Haoqi here.
Yeah. Yeah. Thanks for the introduction. Let me see. Oops. Yeah. Thanks for giving me this opportunity to present in this wonderful group. I think this group is really suitable. Oh, sorry. I mean, my presentation is really suitable for this group because the focus is to look at the brain health and also health in general. And particularly, my talk is about using sleep as a window into health. So basically, what we did is to predict different, probability of future incident outcomes based on sleep, just to see how different people with, for example, high risk of stroke, how does their sleep is different compared to other people. Yeah. So as a very quick background of sleep, so basically we measure in the lab, in the sleep clinic, we measure sleep from 6 channels over the head. So and from the 6 channels, we can get 6 signals. So then usually we represent these signals into this frequency domain so that we can show how does the frequency change over time. So here, x-axis is time from 9 p.m. to the other day, 5 a.m. And then we have 6 panels, and the y-axis shows the frequency. And we have some, as another background, we usually refer to different frequency ranges. For example, 1 to 4 hertz, it's called delta band, and 4 to 8, it's theta band. 8 to 12, it's alpha band, and 11 to 15, some people call it sigma band, some people call it spindle band. So basically, we want to look at how does the sleep, especially how does the sleep signal, the EEG signals look like for and how much information does it contain? How much is it associated with the future outcome? So our key idea is that sleep is a window, and I think it's a bidirectional window that itself reflects multiple aspects of health, including brain health, mental health, and even heart health. And if there's some disturbance in brain health, mental health, or heart health, it will in turn disturb sleep. So I think there is some kind of feedback loop between these two, so that sleep is, in our perspective, a perfect tool that allow us to look into these health issues. Yeah. So our key research question is, does sleep contain information about health? So of course, health is a super general word. So later we will see that we are looking at specifically 11 outcomes that covers brain health, mental health, and also heart health. So just some other background. Many of you know that sleep has multiple functions, including those, the very classic view of sleep consolidate memory. And also sleep helps boosting the immune system. And also, during development, their sleep also helps to regulate hormones. And also recently people got to know that, people hypothesize, and there's some strong evidence that during sleep, brain is trying to clear its metabolic waste. And those wastes are associated with neurodegenerative diseases. And what's more interesting is that, sleep has so many important functions, and yet it's very easy to measure at home. So there is a recent publication showing that some home device, when they compare it to the signals from the clinic level devices, so they are very similar. So in specific, here is a figure showing the black signal. I think it's from a device called Dream. It's measured at home. And then the one below, the red one, it's from the PSG, the clinic standard recording. And in that paper, they show that, they put these two devices at the same person, and then they record the signals at the same time. And it's relatively, qualitatively similar. And they also did a lot of comparisons about sleep parameters, for example, sleep latency, and also, the percentage of N1, N2, N3, and REM, they are all similar. So, so why sleep as a window? So basically it has multiple functions, very important functions. It's a bidirectional window, associated with health. And also, it's also easy to measure at home. So just go a step further. So we have said that sleep is a window into health. So here I show you two examples. One is good sleeper, and on the other slide is bad sleeper. There are some interesting things here. For example, this particular person has a lot of N3, which means deep sleep. And during deep sleep, you can see this very dark color power, spectral power at low frequency range. And also during the other sleep stage called N2, there's a lot of strong power, this light band between 10 and 15 hertz. So this is commonly referred as the sigma or spindle band, which is a reflection of how many spindles it is. From this good sleeper picture, you can see there's a lot of information indicating that the sleep is intact. The sleep is very qualitatively speaking, very good in terms of it has all the necessary features to be defined as a good sleep. And then the other one, bad sleeper, as you can see, the sleep stage, most times it's at awake, and then the sleep also is very fragmented. And if you still remember the previous figure, the previous one has very bright band from 10 to 15 hertz. And this one, even during N2, there is no bright band here around 10 to 15 hertz. And also this person has no deep sleep, no N3. So looks like, sleep contains a lot of interesting and rich information about at least the quality of sleep. And then does the quality of sleep relates to brain health, to heart health, to mental health? That is the thing we want to see. So more specifically about study design. So we have this, we are using this MGH data set, and it's a single center retrospective cross-sectional study. And there are a few inclusion criteria. And the most important one is like we are looking at the diagnostic PhDs so that there is no intervention during sleep. And also we are only looking at the adult population because right now for example, we focus on neurodegenerative disease and also heart disease. They are much more relevant at old ages. And then more specifically, we are looking at 11 diseases, or you can call outcomes. So we have some of them associated with the brain, and some of them associated with the cardiovascular system, and also some of them about the mental health. And also death is one of our outcomes. So basically we got from this diagram, there's at the beginning about 18,000, and then finally we got about half of them, 9,000 patients. Yeah, some of them have multiple PhDs, but they are all diagnostic PhDs. Just a very quick view of the demographics. The age is about 51 years old, about the same male versus female. 75 white people, non-Hispanic 85. Yeah, just a very quick overview. And then we can look at, for example, these so-called Kaplan-Meier curves. So these curves, they summarize over the time, how does the people get into, for example, ischemic stroke. Like the y-axis is the probability of not yet have, for example, ischemic stroke. So this probability will drop over time, and the speed of drop it's related to, it's a general description of how fast does people fall into this state. So if you compare these two figures, and the y-axis of these two figures are the same. So this means that, for example, 12 years since the sleep recording, 12 years after the sleep recording, there is about, let me see, 2% of people who got ischemic stroke. And on the other hand, there's about, I think it's less than 1% of people who got ischemic stroke. Oh, sorry, the left one, sorry. The left one is for intracranial hemorrhage, and the right one is for ischemic stroke. And similarly, we can get these same plots, for example, for the combination of MCI and dementia. So if you still remember the previous figures, these drop, it's about 8%, which is higher than the previous figures. I think it's a, it's a general description of what is the incidence of this disease. So MCI plus dementia is about 8% after 12 years since the sleep recording. And then for dementia, I think this figure is cut somehow, but it's relatively, let me see, it's about 3% at 12th year. And we also summarize these Kaplan-Meier curves for other outcomes. For example, for hypertension, of course, that is I assume is the most common outcome, most common disease. And the circle indicates that this y-axis starts from 0.75, so that this drop is actually, they are on a different scale compared to other figures. But if you read carefully, it's about 11 or 12% for hypertension. And then we also, the other interesting figure is for death. So here, the right top corner, so death at 12 years since the sleep recording, it's about 10% of the people who unfortunately died at 12th year. So these are the general, description of the 11 outcomes in our cohort. So we used survival analysis with a competing risk Cox proportional hazard model, so that, because everyone has two outcomes, either go to the, for example, ischemic stroke, and then unfortunately died, or did not have the outcome and directly died. So of course, this occurs for a small percent of population, but this is the pathway that they eventually will follow. So this kind of competing risk Cox model enables us to quantify, enable us to predict the incidence of outcome. And more specifically, we can predict time to event, or we can predict the curve that goes down with time for each individual subject. So more specifically here, we see three colors, so red, black, and blue. And for each color, we have a solid line and a dashed line. So the dashed line represents a result from a something called non-parametric method. So the dashed line, it's more like a ground truth. And the solid line is from the Cox model we just mentioned, so that we are just to making sure that these two methods, they are consistent with each other. So here we focus more on the solid line, and there are three colors. And the middle one, the black one, are those people who have the risk equal to the mean of the population. So this black curve is like the average trend in the population. And then suppose someone has a higher risk and that a 1 standard deviation higher risk, that will increase that will make the curve go higher. That is the red curve. And then vice versa for the blue one, that's the people who have 1 standard deviation lower risk compared to the mean. So just to understand this figure, so this basically tells us on average, on average plus 1 standard deviation, on average minus 1 standard deviation, how does the risk change with time? And if you still remember the Kaplan-Meier curve from the beginning, that curve is going down while this curve is going up. So they are showing different things. This one is called a cumulative incidence curve, so that the y-axis is the probability of having that outcome. So that will go up over time, and the higher it's the worse. The lower, the better. So basically, we can compute the hazard ratio of these for different outcomes. I didn't put the exact numbers here. I think, for example, the first figure, the risk ratio for between red versus black and black versus blue, it's about 3 times. So the hazard ratio is about 3 times. And also you can see this curve looks like a step, staircase shape. And this is particularly obvious for ICH, but then for MCI plus dementia, it's a bit more continuous. That just indicates that there are more people who got MCI or dementia, so that those steps, they look smaller, closer, so that overall it looks smoother. Yeah, so basically we can see that indeed, if we change the risk, then there is indeed a difference in the incidence. And this risk is a weighted sum of many sleep features plus age and sex. So let me see. Yeah, so you can see these different outcomes, the same kind of curve for different outcomes. And indeed, there is a big difference between these 3 colors. And this is for bipolar disorder and depression. I think for most of the outcomes, the hazard ratio is about 3 times. I think for MCI and dementia, let me go back a bit. I think for this one, it's a bit higher. And we also compute a C index. So C index is a very commonly measured metric for survival analysis. It just tells you how well, for example, if I predict person A to have higher risk than person B, does person A have shorter time to outcome compared to person B? So like the percent, we call this kind of case as concordant, so that you can compute the fraction of concordant pairs among all possible pairs. So I think there is a very interesting thing that actually, based on age and sex alone, we can achieve pretty good performances, and then adding sleep features in different forms, for example, using 5 stages or combining, all the N1, N2, N3 into non-REM. As you can see, there is some improvement, but I think this is the ongoing work that we need to really see if this difference is meaningful or not. So, but at least I think using sleep features consistently across all different outcomes gives improvement over the baseline model. And finally, I just want to show 3 examples. So this is the person who has low risk of incidence intracranial hemorrhage. And this is exactly the person who I said it's good sleeper. I took that as good sleeper. It's the person who has very low risk of incidence ICH. And then people who have medium risk of ICH, and very quickly, people have very high risk of ICH. So the low and high risk, I was copy paste them into the good and bad sleepers. So they agree with our visual inspection, and indeed their incidence is indeed different. And finally, I have 3 discussion points, but I think there's only 3 minutes, so I want to skip this slide and let everyone ask the question.
Thank you, Haoqi.
Okay.
So,
yeah.
I guess questions in the chat, or that's probably the most orderly way. See, so there's a question from Dr. Kim. He says, excellent talk. I wonder if the abnormal sleep pattern you detected can cause future pathogenic incidents or simply suggest early stage brain disturbances by the pathogenic condition in the future. I think he's asking about cause and effect.
Yeah, that's a very important question. I think all what we do is by association, right? Because we know that there is a bidirectional relationship between these two. So that it, by just doing survival analysis, it's a way to look at their association. If we were really to do causal, like, to ask whether bad sleep causes bad brain health, I think we need, some intervention on the same patient done longitudinally. That's also one of my discussion points here, that, if we could do some kind of intervention targeting at sleep, that will really answer the causal question. I think this is ongoing work, very exciting ongoing work. I hope one day I can answer you confidently.
Looks like we have another question from Dimitri. Great talk. I was wondering why do you see a saturation in the Kaplan-Meier curves? So what is it that happens at around 10 years that makes things level off?
Let me see. I think it's likely to be people are dead after many years because as you see, there is a competing risk. People can either go to have developed that outcome or die eventually. Of course, everyone eventually. But I would say it's probably due to that. Like if nobody died, they have enough time to develop the outcome. I would say it will keep going down. But the reality is there's competing risk.
There's also, it looks like there's very few people left out that far, I think, right? Most of them are censored. So I wouldn't probably worry too much about the shape there. It's a little less reliable statistically.